Welcome to this short presentation from neurexpert, experts in applying electrophysiology to drug discovery and the understanding of drug targets and neuronal function. This video describes human EEG recording and how an isolated brain slice preparation can be used to model this activity for the purposes of drug discovery. In humans, the electroencephalogram is a non-invasive method to record electrical brain activity. This method is extensively used for research and diagnostic purposes in a wide range of neurological and psychiatric conditions. Such recordings can show specific rhythmic activity that can be characteristic markers of behavioural states or disease conditions, and an example is shown in the chart panel on the right. For experimental purposes, it is possible to make brain slices that are then kept in a physiological state in a recording chamber. Here it is then possible to record electrical activity directly from individual cells or groups of cells in specific areas of the brain. Shown here is a recording from the CA1 area of the hippocampus, an area of the brain that is known to be involved in learning and memory. Under the appropriate conditions, it is possible to record rhythmic or oscillatory activity from the brain slice. The green trace shows rhythmic activity in the 30 to 40 Hz frequency range. This frequency range is often referred to as the gamma frequency range in EEG recordings and is sometimes associated with attention and/or sensory processing. Above the green trace is shown the distribution of power in the signal over different frequencies. In the spectrum, low power is shown as black, with increasing power arbitrarily represented by red, orange, yellow, white and blue colours. It is clear to see that the maximum signal power is in the 30 to 40 Hz range, as shown by the arrow. This power distribution, or spectrum, can be quantified. Shown here is a histogram illustrating the power at different frequencies in this recording. Note peak activity is in the 30 to 40 Hz range, as shown by the arrow. This activity in the slice can thus be considered to model human EEG activity in these conditions. It is possible to investigate the effects of endogenous neurochemicals or drug candidates on this electrical activity. Shown here is activity recorded from the same brain slice, but in the presence of a drug that is known to modulate excitatory synaptic transmission. This drug causes a substantial change in the rhythmic activity. In this example, there is a shift in peak in the gamma range to approximately 30 Hz, and also a large increase in power in the low frequency range below 15 Hz. This frequency range corresponds to the theta and alpha ranges of the EEG. The theta and alpha ranges can be related to different sleep states. It is thus possible to evaluate drug action in an in vitro system and relate this to brain activity that can be recorded from humans under different physiological and pathological conditions. This makes the recording of oscillatory activity in brain slices an important tool to translate from the laboratory to the clinical situation. We hope you enjoyed this short video. For more information on this assay or any of our other assays and services, please contact us at www.neurexpert.com or email us at info@neurexpert.com.